Good morning, ladies and gentlemen. My name is Trevor, and on the behalf of the Pharmacy Academic Sehat Mandiri Sidoarjo, I would like to welcome all of you to the international webinar of Afamedis International Conference on Health and Pharmaceutical Development (AIC) towards new normal era in 2021. We would like to extend very special thank you to the Deputy Head Region of Sidoarjo, Mr. Subandi Bachelor of Law, and the Director of Academy Pharmacy Sehat Mandiri Sidoarjo, Mr. Apoteker Andri Priyo Herianto, Bachelor of Pharmacy, Magister of Science, and our speakers today, Dr. Marlon from the Universitas Federal Rio de Janeiro, Brazil, Mr. João Vitor Roca Reis from the Universitas Federal Rio de Janeiro, Brazil, and Dr. Apoteker Cikra Iqbal, Magister of Science from Academy Pharmacy Sehat Mandiri Sidoarjo, and lastly, for all of the participants in today's event. Ladies and gentlemen, at this time, we ask you all please rise for the playing in of Indonesia Raya and Academy Pharmacy Sehat Mandiri Sidoarjo Trans. You need to be sitting. Next is the welcome dinner by the Deputy Head Regent of Sidoarjo, Mr. Subandi, Chancellor of Law. Mr. Subandi, Chancellor of Law, the time is yours. Kepada Bapak Subandi, kepada Ratu Sultan Muhammad untuk menunggu.
Minggat. Masa aku dulu.
I'm so sorry that is out. And this session is a welcome by the Director of Academic Informasi Kesehatan Mandiri Sidoarjo. Ladies and gentlemen, Mr. Apotheker Andre Priyo Herianto, Bachelor of Pharmacy, Magister of Science.
Oke, terima kasih. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera untuk kita semua. Untuk suara saya apa bisa sudah terdengar ya, Mbak MC?
Sudah, Pak Andre.
Ya. Oke, baik. Terima kasih. Yang pertama, selamat datang dan selamat bergabung kepada Dr. Marlon dari Associate Professor Universitas Federal Rio de Janeiro, Brasil, yang pada pagi hari ini bersedia dan berkenan untuk berkolaborasi, berbagi ilmu, dan mungkin suatu saat nanti bisa melibatkan kami di sini untuk penelitian dan pengembangan terkait dengan potensi alam yang kita miliki di sini. Yang kedua, saya sampaikan juga terima kasih kepada Mr. Joe Victor Rosa Reis, Graduate Student of Pharmacy, yang juga kali ini akan berperan sebagai narasumber yang kedua. Kemudian yang saya hormati Bu Dr. Cikra Ikhda, yang hari ini juga akan bertindak sebagai narasumber ketiga, sekaligus Wadir 1 Akademi Farmasi Mitra Sehat Mandiri Sidoarjo. Kemudian yang kami hormati Pak Ira selaku Wakil Direktur Humas atau Bidang Kemahasiswaan. Yang kami hormati para peserta seminar baik yang dari Akademi Farmasi Mitra Sehat Mandiri Sidoarjo maupun yang dari kampus yang lain, serta yang kami hormati para tamu undangan, terlebih pada Bapak Haji Supandi, Sarjana Hukum, yang sedianya nanti pukul 9 baru bisa membuka kegiatan ini secara resmi, karena mengingat pagi ini masih dilaksanakan apel di Pendopo Bupati sampai pukul 8.45, sehingga nanti pukul 9 baru bisa bergabung dengan kita semua. Alhamdulillah, puji syukur kehadirat Allah Subhanahu wa Ta'ala, yang mana sampai pada pagi hari ini kita semua masih diberikan semangat sehat, motivasi yang tinggi, kemudian kesehatan yang luar biasa di tengah pandemi COVID-19 yang sampai detik ini masih belum pergi dari kita semua. Semoga kita senantiasa diberikan perlindungan, diberikan kesehatan, diberikan kemudahan, dan diberikan kelancaran dalam segala hal dimanapun dan kapanpun kita berada. Para peserta seminar yang kami hormati, pagi hari ini merupakan salah satu bentuk upaya dari kita, dari Akfar Mitra Sehat Mandiri Sidoarjo, di bawah Koordinator Bu Jikra selaku Wakil Direktur Bidang Akademik, sebagai bentuk partisipasi kita untuk memikirkan dan mengembangkan tentang potensi alam kita untuk supaya berperan dalam pengobatan TBC. Mengingat TBC sekarang ini saya rasa Indonesia mungkin ada di peringkat 5 besar dunia. Terlebih Sidoarjo kemarin sudah sempat ada data yang menunjukkan bahwasanya Sidoarjo ini juga saya kira masih dalam tahap yang warning juga terkait dengan TB. Sehingga dengan kegiatan hari ini ada harapan salah satu upaya solusi dan langkah-langkah yang terbaik yang bisa kita lakukan sebagai bentuk kontribusi kita sebagai lembaga pendidikan terhadap pengembangan dan pemeliharaan kesehatan maupun peningkatan kesejahteraan masyarakat kita khususnya di Kabupaten Sidoarjo. Kami juga berharap besar kepada Dr. Marlon, kemudian Mr. Joe Victor untuk selalu mendampingi kami, membimbing kami, kemudian bertukar ilmu, kemudian bertukar pengalaman dalam rangka peningkatan keilmuan sesuai dengan bidang akademik kami. Semoga acara pada pagi hari ini nanti dapat diambil manfaatnya, ya. Kemudian saya percaya dan kita percaya tidak ada hal yang sia-sia terkait dengan apa yang sudah kita lakukan pada detik ini. Sekecil apapun itu pasti akan ada manfaat dan kebermanfaatannya untuk kita ke depan. Yang terakhir saya sampaikan mohon maaf kepada Dr. Marlon maupun Mr. Jauh serta semua peserta kegiatan atas segala hal yang mungkin kurang berkenan atau ada hal teknis yang mungkin masih dalam tahap kurang baik atau kurang sempurna. Semoga ke depan kami dapat memperbaiki teknis maupun non teknis dalam pelaksanaan seminar ini sehingga segala satunya berjalan dengan lebih baik. Terakhir sebelum saya tutup, saya sampaikan terima kasih dan luar biasa kepada panitia maupun tim IT yang sudah berupaya luar biasa berkolaborasi untuk menghadirkan kegiatan seminar internasional ini yang sudah lama kita agendakan. Akhirnya pada pagi hari ini dapat kita laksanakan dengan baik dan lancar dan nanti juga akan dibuka secara resmi oleh Bapak Wakil Bupati Sidoarjo pada pukul 9. Mungkin itu yang dapat saya sampaikan. Sekali lagi terima kasih dan kami berharap suatu saat nanti Dr. Marlon maupun Mr. Joe Victor dapat berkunjung ke negara kami, dapat hadir ke Mitra Sehat Mandiri Sidoarjo untuk bertatap muka secara langsung dan kami akan mengenalkan Anda berdua terkait dengan budaya kami di Indonesia yang memang negeri seribu budaya, negeri banyak suku, negeri banyak pulau, dan negeri banyak tanaman alam yang bisa kita eksplor untuk kemajuan farmasi di bidang obat. Terima kasih, semoga jalannya berjalan dengan baik dan dapat memberikan manfaat pada kita semua. Billahi taufik walhidayah. Assalamualaikum warahmatullahi wabarakatuh. Saya kembalikan ke Mbak MC, silakan.
Thank you, Mr. Apotheker and Ibu Herian, Professor of Pharmacy, Magister of Science. for your briefing today. And the third agenda is introducing Afamedis Mandiri Sidoarjo, Mr. Heras Widyoko, Magister of Management. The time is yours.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera bagi kita semua. Mohon izin saya share screen terkait dengan video profil Akfar Mitra Sehat Mandiri Sidoarjo. Lambat. Kapan pasangnya, Pak? Nggak ada suaranya, Pak. Oh, lagi betul aku. Nggak ada. Oh, siapa ono? Baru bisa. Iya.
Bapak Muhammad Ali.
Well, ladies and gentlemen, it is right along with the call of today's event. It is my pleasure to introduce our moderator, Ms. Erna Fitriani, Magister of Science.
Sama
Ms.
dengan
Erna is elected to the Department of Pharmaceutical Chemistry at Akademi Farmasi Putra Sehat Mandiri Sidoarjo. Ms. Erna Fitriani finished her Magister of Science at Airlangga University. Her research focused on developing sensors in the field of pharmaceutical chemistry. Please give a warm welcome to Mr. Erna Fitriani, Magister of Science.
Okay, thank you to Miss Phoebe, and Assalamualaikum warahmatullahi wabarakatuh, and good morning to all of you. The Excellency, the Deputy Head Regent of Sidoarjo, Mr. Haji Subandi, Bachelor of Law, and our Director of Akademi Farmasi Mitra Sehat Mandiri Sidoarjo, Mr. Apoteker Andri Priyo Herianto, Bachelor of Pharm, Magister of Science, and the honorable participants of this international webinar of Afamedis International Conference on Health and Pharmaceutical Development EIC today. And I'm Erna, your moderator for this session. And I'm very pleased to see all of you and welcome you to this conference. So for these sessions, we will have 3 speakers. And the first speaker is Dr. Marlon from the Universidad Federal Rio de Janeiro, Brazil. And our second speaker is Mr. Joao from the Universidad Federal Rio de Janeiro, Brazil. And the third speaker is Dr. Cikra Ida Nurhamida Savitri from Akademi Farmasi Mitra Sehat Sidoarjo. And because of something wrong, and I'm so sorry, our first speaker is Dr. Cikra Ida Nurhamida Savitri from Akademi Farmasi Mitra Sehat Mandiri Sidoarjo. At the first, let me to deliver the curriculum vitae of Dr. Cikra Ida from Akademi Farmasi Mitra Sehat Mandiri Sidoarjo.
Nanti pertanyaannya lewat Zoom dulu aja.
And this is the profile about Dr. Cikra, and Dr. Cikra received her doctorate in Pharmaceutical Sciences at Gajah Mada University, Indonesia, and she gets her Master of Science at Setia Budi Surakarta University, that focus on pharmaceutical science, and she gets her bachelor degree at University of Jember, Indonesia. And she also published some papers that focus on natural product, tuberculosis synthesis, and cosmetic. And Dr. Chikra also one of the WHO researchers in Indonesia who is developing a vaccine against the coronavirus. And without further ado, Dr. Chikra, this meeting is yours.
Okay, thank you, Miss Erna. Can you hear me very clearly or maybe slowly?
Yes, Dr. Chikra,
Okay.
very clearly.
Okay, thank you. Mr. Rizal, can you show my presentation in the Zoom room? Sorry. Mr. Rizal, can you present? Okay. I'm so sorry because something terrible in the connection. So Mr. Rizal will show my presentation. And okay, thank you. Okay, good. Well, thank you for my times and Mr. Marlon, Mr. Dr. Marlon and Joe. I will first to present my paper. And well, ladies and gentlemen, I would like sharing about 2 decades of TB drug discovery in the world. Okay, the first presentation, sorry. I'm sorry, maybe Mr. Erna can show the presentation. Belum. Untung belum.
Okay, ladies and gentlemen, because our region of Sidoarjo, Mr. Haji Subandi, Bachelor of Law is come. And now I return to the MC at first. Thank you, Miss Phoebe.
Please welcome Mr. Haji Subandi. The floor of the time is yours.
Selamat datang Bapak Wakil Bupati Sidoarjo di Akademi Farmasi Mitra Sehat Mandiri Sidoarjo.
Selamat datang Ibu, selamat pagi.
Selamat pagi, Pak.
Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi dan salam sejahtera bagi kita semua. Bismillah,
Selamat
alhamdulillah.
pagi.
La haula wa quwwata illa billah, amma ba'du. Yang hormati Direktur Akademi Farmasi Mitra Sehat Mandiri Sidoarjo. Yang hormati Dr. Marlon Ikrodin de Arijo sebagai pembicara pertama di Universitas Federal de Janeiro. Yang hormati Jauh Victor Rogaroi sebagai pembicara kedua Universitas Federal de Janeiro. Yang hormati Dr. Apoteker Citra Ika Nur SHS, B.Pharm MSc M Biotech sebagai pembicara ketiga dari Akademi Farmasi Mitra Sehat Mandiri Sidoarjo dan semua agensi seminar pada hari ini. Puji dan syukur aturkan pada Allah Ta'ala pada kesempatan hari ini kita bersama-sama bisa menghadiri dalam acara kegiatan seminar Mitra Mandiri Sehat dengan keadaan sehat walafiat, tidak ada halangan suatu apapun. Hadirin sekalian, senang kali saya dapat menyambut Anda di Konferensi Internasional Akademis tentang Perkembangan Kesehatan dan Formasi di Sidoarjo, Jawa Timur, Indonesia. Saya sangat berterima kasih kepada para panitia yang telah menyelenggarakan acara yang luar biasa ini. Saya merasa terhormat secara khusus menyambut perwakilan dari masing-masing universitas, institusi, dan organisasi berpartisipasi dalam konferensi ini. Dalam konferensi pada hari ini, kami mempelajari tuberkulosis dan penemuan obat hingga 2 dekade. Indonesia merupakan negara yang memiliki keanekaragaman hayati yang besar di dunia. Bersama Brasil, Sidoarjo juga memiliki kasus tuberkulosis yang juga diperparah dengan keberadaan penderita HIV. Salah satu pencegahan khusus tuberkulosis juga dapat bermanfaat obat-obatan herbal atau obat alam yang di sekitar kita. Selain itu perlu juga diberikan pemahaman yang lebih mendalam tentang perkembangan pengobatan tuberkulosis, di mana belum ada obat baru, padahal kasus retensi sudah mulai meningkat dan jenis resisten baru. Saya berharap konferensi ini dapat memberikan kesempatan untuk bagi hal ilmiah dalam kemitraan dengan Brasil. Atas nama Bupati Sidoarjo, saya ucapkan terima kasih atas terselenggaranya kegiatan ini. Semoga berdampak positif bagi para peserta, khususnya Kabupaten Sidoarjo. Semoga seminar ini dapat membantu Anda membangun pengalaman ilmu pengetahuan kemitraan. Bapak Ibu yang hormati, alhamdulillah, atas nama pemerintah Kabupaten Sidoarjo dengan diselenggarakannya kegiatan-kegiatan, mudah-mudahan nanti ini bisa membangun suatu keharmonisan atau tukar pengalaman antara rumah sakit mitra keluarga yang selama ini dipercaya di Rumah Sakit Sidoarjo, di pemerintah Sidoarjo. Mudah-mudahan nanti membawa kemaslahatan, membawa kebaikan, kerjasama dengan baik dengan luar negeri. Ini juga nanti terbangun dengan rumah sakit yang lainnya. Dengan adanya seperti ini juga untuk menambah ilmu kita, inovasi kita, di mana antara rumah sakit mitra keluarga ini bisa membangun kemandirian dan kerjasama dengan baik, apalagi dengan dari negara Eropa. Ini alhamdulillah luar biasa. Mudah-mudahan saya memberikan apresiasi yang tinggi kepada Direktur Rumah Sakit sebagai pengelola Rumah Sakit Mitra Keluarga. Mudah-mudahan nanti ke depan terus eksis, terus berkembang. Mudah-mudahan nanti rumah sakit ini sebagai rumah sakit rujukan yang luar biasa dari rumah sakit yang lainnya. Bapak Ibu yang hormati, saya sudah bisa melihat potensi besar dari para peserta. Saya berharap melalui acara ini, kita semua bisa belajar menjadikan pemikir yang baik dan kritis. Nah, saya tidak ingin terlalu banyak menyita waktu Anda. Saya perlu menyerahkan Master Company pembawa acara kami hari ini untuk memperkenalkan pembicaraan tamu kepada Anda semua. Sekali lagi saya ucapkan selamat datang di seminar ini. Senang melihat banyak dari Anda di sini. Sekali lagi selamat datang dan terima kasih semuanya yang ikut dalam kegiatan seminar pada pagi hari ini. Itu saja yang perlu kita sampaikan, yang lebihnya terima kasih. Wallahu muwaffiq ila aqwamit thoriq. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you, Mr. Sukai. A round of applause for your greeting in this Afamedis International Conference on Health and Pharmaceutical Development (AIC) towards new normal era in 2021. Now, I return to the moderator, Ms. Erna Fitriani, Magister of Science.
Okay, thank you for Miss Phoebe. And now we will continue about Dr. Cikra's presentation. And his research also focus on tuberculosis. And Dr. Cikra, this time is yours. It only 30 to 45 minutes.
Okay, Ms. Erna, can you show my presentation? Okay, well, okay, we come back again. Okay, well, ladies and gentlemen, I would like to share about 2 decades of TB drugs discovery in the world. As we know, on 24th of March ago is Tuberculosis Day. So happy Tuberculosis Day for all. During the pandemic, tuberculosis wasn't spotlight and said it focused as the government and society focus on COVID-19. So please first PowerPoint. Okay. Okay. We know in the figure or in the picture based on World Health Organization report 2021, most people who developed tuberculosis in 2019 were in the region of South Asia and then Africa and the Western Pacific, with small percentages in the Eastern Mediterranean, Americas, and Europe. And a country, okay, and a country account for 2/3 of the global total. India, Indonesia, our country, China, the Philippines, Pakistan, Nigeria, Bangladesh, and South Africa. The other 22 other countries in World Health Organization's list of 20 high TB burden countries account the global total. And the tuberculosis incidence rate at the national level varies from less than 5 to more than 500 new and relapse cases per 100,000 population per year. And in 2019, of tuberculosis. In previously treated cases were countries in the former Soviet Union Soviet. Okay. In 2014 and 2019, all member states, World Health Organization, and the United Nations commit to ending the TB epidemic through their adoption and World Health Organization and TB strategies and the United Nations Sustainable Development Goals, or CDGs. The strategy and CDGs include milestone and target for large reduction in TB incidence, TB deaths, and costs faced by TB patients and their households. And we know, I'm so sorry, in the previous, still in previous, okay, yes. And we know in the table, we know the target or effort the state political commitment to fight against tuberculosis intensive in 2017 and 2018. World Health Organization Global Ministerial Conference on TB Awareness Organization in November 2017, like in United High Level Posting on Tuberculosis 2018. So we know and many children is infected by tuberculosis. So the strategic for tuberculosis is indeed. Next. Well, the picture show the three colors as blue, red, and orange, like this one. In the blue is first line of tuberculosis drugs, and then red is second line is tuberculosis drugs, and orange is recently approved by Food Drug Approvement. Historicon, streptomycin, and para-aminosalicylic acid among the first clinical antibiotic developed, but saw the activity against Mycobacterium tuberculosis and were followed in rapid succession by isoniazid, pyrazinamide, cycloserine, etinomide, ethambutol, and rifampicin, among others. A number of new potential anti-tuberculosis drug candidates with novel modes of action have been entered in clinical trial in recent years, like delamanid or bedaquiline. These agents most likely be effective against resistant strain. The treatment landscape is beginning to shift with the recent approval by Food and Drug Administration to new tuberculosis drugs, and then bedaquiline and delamanid. Also, the pipeline of potential new treatment has been fulfilled with several compounds in resistant Mycobacterium tuberculosis bacteria. An additional new chemical entity is also under development, and the already existing drugs with their target mode of treatment, as well as new probable anti-tuberculosis drugs, muitis, which is the reason the pipeline has summarized in this our review now. Next. So we know emerging challenge in TB treatment. The emergence of extensively drug-resistant strains of Mycobacterium tuberculosis is so hygiene, so challenges TB treatment program in several other countries and even raises the possibility of a return to a situation akin to pre-antibiotic TB era. At present, multiple drug-resistant tuberculosis is treated by combination of 8 to 10 drugs with therapy lasting up to 18 until 24 months. Only 4 of these drugs with actually development to treat tuberculosis. Such suboptimal therapy leads to almost 20% of multiple drug-resistant tuberculosis patients experiencing treatment failure. The one is the treatment options for extensively drug-resistant tuberculosis are very limited, as extensively drug-resistant tuberculosis, bacilli are resistant not only to isoniazid and rifampicin, but also of fluoroquinolone and injectables such as aminoglycoside. And in addition, there are serious side effects with most multiple drug-resistant tuberculosis and extensively drug-resistant tuberculosis drugs. Includes nephrotoxicity, ototoxicity with aminoglycoside, hepatotoxicity with ethinoamide, and dyslipidemia with gantifloxacin. Thus, the current situation necessity the immediate identification of new scaffolds. New scaffold that can address emerging resistance and also demand the conduct of appropriate clinical trial, as historical very few clinical studies have been performed to evaluate the efficacy of drugs of multiple drug resistant tuberculosis or extensively drug resistant tuberculosis patient groups. So to improving the diagnostic with wider coverage of drug susceptibility testing, will also have to address the high mortality of multiple drug resistant or extensively drug resistant and curb the emerging of resistance. And third, the frequent co-infection of tuberculosis in human immunodeficiency virus patient further complete. And the fourth, the main interaction between human immunodeficiency virus and tuberculosis anti-infectives is rifampicin induced increased expression of cytochrome P450 oxide system. This cytochrome-induced result on increased metabolism and decreased therapeutic concentration of many co-medication, such as human immunodeficiency virus protease inhibitors. So, even in the presence cytochrome inhibitors such as ritonavir, normal tox level of various classes of protease inhibitor cannot be rescued, and consequently, standard protease inhibitor regimen, whether booster or not, cannot be given with rifampicin. So the only treatment for human immunodeficiency virus infected in tuberculosis patient with minimal drug-drug interaction are non-nucleoside reverse transcript inhibitor or NNRTI. So, however, there are fewer options for patients with NNRIT resistant mutation, and therefore new chemistry approach are being to identify new rifampicin or the new drugs. Next. Okay. The several anti-tuberculosis drugs currently in use, have been discovered based on the learning that follow the introduction of each the compound to drug regimen. The data have with several pointers, while each successful drugs was discovered building on the state of the knowledge. The newer drugs themselves also help in the further understanding of the pathogen biology. This is a reflected current set of drugs that are in late stage development or have been recently introduced into the anti-tuberculosis regimen. The following groups like serendipitous drug discovery, modification of drug scaffolds, and target-based screening, and the last phenotypic screening. So now we know the 4 anti-tuberculosis drugs is currently progresses into decade tuberculosis development. Next. Okay, the first serendipitous drugs discovery. Okay, the serendipitous drug discovery, early chemotherapy. We know about chemotherapy. The chemotherapy is the first successful in cure an infectious disease. Is indeed the discovery and design of the first line therapy for the treatment of tuberculosis. The design and development of which was complete in the 1960s. Even today, its regimen is therapy of choice for treat the tuberculosis. For sensitive tuberculosis, the drug is the first line treatment for Treatment for tuberculosis like isoniazid, rifampicin, pyrazinamide, and ethambutal become anti-tuberculosis drugs based on their activity of Mycobacterium tuberculosis cells in vitro, followed by testing in animal models and then rapid introduction into human. So this progression with the medical needs as chemotherapy as before there's were drugs discovery. So it is more interesting because we know about the serendipitous drug discovery have four key learning from this pioneer are as follows. The first, in vitro MIC or minimum inhibitory concentration was insufficient to predict efficacy in human. So we never know about the effectivity. And the secondly, requirement for a combination therapy of drugs. As we know, like combination rifampicin, isoniazid, and extra. And the third, the best regimen required 6 months of treatment to achieve cure. So we know the 6 months is more for a long period. And the last, each of the drugs in the combination had a unique role to play in leading to the cure. So we know the cure was defined the lack of relapse. The last 2 points remain today the biggest challenge in finding development novel combination. Next. Okay, the second about modification of drug scaffold. We know maybe more than 4 modification of drug, but I think we can learn 4. The first, and the first about nitroimidazole. I think nitroimidazole used to treat anaerobic bacteria and parasitic infections, and then represent another established scaffold for which synthetic modification have been introduced to increase their antimicrobacterial potential. An interesting feature of nitroimidazole relates to their unique mechanism of action, mimicking host defense strategies by produce microbicid, I think antimicrobicid, and then such as nitric oxide and their reactive nitrogen intermediate, which is damage multiple targets include respiratory chain cytochrome oxidase. The target specificity such as mechanism of action is absent in mammalian cells, but present in Mycobacterium tuberculosis. So to candidate from this class like PA824 and OPZ97883 are currently in clinical studies and may potential shorten treatment duration as this mechanism of action. And then next slide. Like this, nitroimidazole as starting point. This one, nitroimidazole, we can get metronidazole. We know that metronidazole is antibiotic. Then present in the pyrazinamide and then can abroad in the delamanid. Yeah, delamanid is one of new anti-tuberculosis drug from World Health Organization therapies. So we can know delamanid and bedaquiline is the new anti-tuberculosis drug in 10 years ago. Okay, next. Okay, the third is target-based screening. Like the figure, the Bull's A representation of the current clinical pipeline for tuberculosis. Each drug candidate is shown with current clinical phase of development along with the target family, like this one. Okay, and then TMC 207 is in 2 trials for multiple drug-resistant tuberculosis. In phase 2 trial for sensitive tuberculosis, and then the structure has not been disclosed with Delamanid, a new drug application. So we know about the new structure, more different with the first line of tuberculosis drugs like rifampicin and extra. Okay, the identification of new tuberculosis drug targets have been driven largely by the availability of the genome sequence of Mycobacterium tuberculosis. But unfortunately, this approach has yet to lead the identification of new drug candidates. Genome-derived target-based approach have had a little success in the antibacterial therapeutic area in general. So the essentiality of the target of replication may be prerequisite, but it does not ensure its drugability. For many essential targets, we are unable to identify specific inhibitor with drug-like properties. The reason success the whole cell screening approach is particularly like the identification of new tuberculosis drug candidates such as diarylquinoline. Diarylquinoline we call TMC207, which is target ATP synthesis, and benzotiazine or BT043, which target essential cell well arabinine synthesis. And we know arabinine synthesis is well designed in the Mycobacterium tuberculosis. Interesting feature of both molecule is that they target membrane-associated protein that may be more easily accessibility to drugs from the periplasmic space. So we know that is the target binding site from Mycobacterium tuberculosis expose the periplasm. So we know, and to some extent may overcome certain issues of mycobacterial membrane permeability. Interestingly, more refined multi-target pathway screens can be initiated to search for inhibitor block validate metabolic or signaling pathway from Mycobacterium tuberculosis cell wall. In this regard, respiratory membrane vesicle of Mycobacterium tuberculosis, which have been grown in variety condition, I think in order to simulate host microenvironment, so can be used to screen drug classes of analog inhibiting respiratory then compound. For example, such a pathway screen could monitor a drug's influence on the first Mycobacterium respiratory chain functions such as ATP synthesis, redox homeostasis, and proton gradient. So, modulating external growth stimuli such as carbon source, micronutrient, or oxygen levels in such an assay result in target respiratory proteome can be used to screen against function the essential during those metabolic states. For instance, ATP synthase in highly down regulation during hypotoxic condition and the inhibition by TMC and more the molecule. Then next. Okay, we will see about protein synthesis inhibitor. The protein synthesis inhibitor, like oxazolidine class compound, it is can treatment for multiple drug resistant and extensively drug resistant tuberculosis. So the protein synthesis inhibitors mechanism in linezolid and like currently a part of the drug regimen for the treatment of multiple drug resistant tuberculosis, extensively drug resistant tuberculosis, and then non-responding tuberculosis case. And then while newer oxazolidines like postisolid and sutasolid have been tested in advanced clinical trial, but now we wait how the result from clinical trials. And then beta-lactam as antimycobacterials, two novel compounds that clinical development from beta-lactam, like leucine, belongs to oxabaryl class of compound and has been shown the leucine tRNA synthase inhibitor, one of enzyme. The compound is currently in clinical trial. And then the SPR720 is a GRB ATPase inhibitor, same enzyme that belong to benzimidazole class, the benzimidazole analog. The molecule is also in phase 2 clinical trial. And then a very recent report shows that SPR720 obtained an orphan in disease status from Food and Drug Administration to treat non-tuberculosis mycobacteria. Next. This one, the molecule of G and SPR720. And we know that chlormuitis, hydroxyl, and then in the SPR like fluor, and then hydroxyl. So we can know about the fluormuitis or chlormuitis or hydroxyl groups is essential groups for anti-tuberculosis agent. Next. Okay, and the third, gyrase inhibitors. Gyrase inhibitors have mechanism action of the fluoroquinolone class like moxifloxacin, levofloxacin, ofloxacin, and gatifloxacin compound. This is a potent inhibitors of the DNR gyrase enzyme and the proven antibacterial. Moxifloxacin now part of the standard regimen to treat tuberculosis resistant like multiple drug resistant, extensively drug resistant. And the last, the AstraZeneca India effort, I think this one is produced from AstraZeneca, and then the target for the new drugs to anti-tuberculosis agent, and follow the two mechanism, and this one is gyrase at the target, and we know that gyrase is one of the favorite target for anti-tuberculosis drug discovery because the gyrase enzyme is essential, and multiple steps involve the mechanistic of the negative supercooling enzyme reaction. So the several steps which have been shown in the inhibiting in Mycobacterium tuberculosis. And then the availability, several crystal structure, the enzyme will help in developing different screening approach as well as embodying structure analog and then identify inhibitor in program. Okay, next. Okay, the last of drug discovery of tuberculosis is phenotype screening. Like see in the phenotypic screening, like in bedaquiline, is one of the new anti-tuberculosis drugs approved in 2020 for the treatment of multiple drug resistance, and then being first FDA approved tuberculosis drugs in more than 4 decades, and then discovered by Johnson & Johnson, and many compound against Mycobacterium stigmatis, other Mycobacterium marino, Mycobacterium tuberculosis, and a compound first described in 2021, like this, at the Interesting Conference by Antimicrobial Agents and Chemotherapy. The target of bedaquiline is the ATP synthase, and we know that is essential part of multiple drug resistant regimen, and several combinations of bedaquiline investigated for their potential to reduce the duration. So bedaquiline is effective for resistance case like multiple drug resistant, extensively drug resistant, but I don't know about totally drug resistant. So bedaquiline is investigated. The next. Okay. And the secondly is benzothiazinone and macozinone. Benzothiazinone discovered the Russian Academy, and the target is decaprenyl phosphoryl ribose oxidase enzyme, yeah, by genetic and by chemical studies. Based on SAR exploration, derived with superior pharmacokinetic as compared that benzothiazinone, and now is phase 2 clinical trial that like macozinone. Next. Next. So sorry. It's loading, I think. Okay. Literature back. Okay, yeah. Azaindole. Azaindole is I get it from literature, and then I like a similar compound with Q, like this one, and the fluoromoids, and then have not have hydroxyl, but have fluoro, and then the compound had an MIC but was not bactericidal. So just inhibiting without killing. Scaffold morphing efforts on this compound, like the TBA, potent for the enzyme DPRE1 inhibitor. And then now is clinical trial phase 2 for TB patient, but the new case for tuberculosis, not multiple drug resistant, also extensively drug resistant. So azaindole is the new anti-tuberculosis for the new case for tuberculosis agent. Next. Okay, and then we know about telacebec. Telacebec or Q203 discovered by Korea, South Korea, I think, inhibits in cytochrome of Mycobacterium tuberculosis. And then the critical point is the electron transport chain. So as we know, the electron transport chain is the good mechanism, and the compound is phase 2 clinical trial. And then the structure of OPZ, we know that OPZ from UK in 2016. The mechanism is carboxyl derivative that inhibit the enzyme DPRE1, same with telacebec. The structure have hydrophobic and hydrophilic residues, but this is like to balance. And then the compound is phase 2 clinical trial too. Next. Okay. This one is the table for phenotypic screening based effort for at AstraZeneca India. So we know that two targets, DPRE1 enzyme and ATP synthase, yes, to target. We know that DPRE1 for the compound benzothiazole, aminoquinoline, piperidine, amida, pyrazolopyridone, and azaindoles, and then the benzoimidazole. And for ATP synthase, like sporamide, imidazopyridine, and diaminoquinazoline. So 3, 4, 5, 6, 7, 8. Each compound is in the phase 2 clinical trial. Next. Okay. The new one about the anti-tuberculosis pipeline. Yeah, we know the pipeline is anti-tuberculosis drugs have evaluation. I think because it is a lead generation approach, the pipeline start by pre-chemotherapy until make the drugs. As the pipeline evolve based on the significant increase the knowledge of biology of Mycobacterium tuberculosis is the more question. Then. Okay, maybe about the combination therapy. As we know, the tuberculosis agent have many combination therapy. But I would say about finding novel combination to change the rifampicin, isoniazid, and extra. The concept for combination is reason the first advocated by the study for using for humans, and then. Recently approved drugs like bedaquiline and delamanid were progressed adds on to cocktail second-line drugs. And we know like pretomanid in combination with bedaquiline and linezolid and approve the treatment for extensively drug-resistant tuberculosis. I think it's all the resistant of tuberculosis, but it's very limited choice. And then the third, can compound active on the same target be compatible in the same combination? It's possible because the two inhibitors bind to different sites on the same target. So the combination will be inhibit two ways, can same or others ways. Okay. So we can conclude that the first, finding new drugs for treatment for tuberculosis to be continue, and the challenge despite increased effort over the last 2 decades, and the lead generation approach for for Mycobacterium tuberculosis drug discovery have undergone several changes. Yeah. And the technological advance mirrors the increased understanding by biology of the pathogen. And the last, interestingly, much of increased understanding is also the consequence of the introduction of newer drugs for treatment of tuberculosis. Okay, I think the last for my presentation, and as we know that many drugs to development to new anti-tuberculosis agent. So we know about the new anti-tuberculosis agent from the synthesis, chemical product, or analog like curcumin analog, azoxazole analog, or indole analog, and the analog extra. So we can know about the new anti-tuberculosis agent. Okay, thank you.
Okay, thank you for Dr. Chikra for your interesting presentation that focused on tuberculosis drugs. And based on her explanation, we can conclude that on the last 2 decades, researchers have a challenge on finding new drugs for the treatment of tuberculosis. Right, Dr. Chikra?
Okay, yeah.
Yeah, and in this session, we have come to question and answer sessions, and we have 10 minutes to 15 minutes to discuss about Dr. Chikra's explanation. Yeah, and to all of the participants, please raise your question in this chat Zoom, or you may raise your hand at this discussion tab. And we have a question, Dr. Chikra, from Mr. Terry Septa. And the question is, well, a patient consume OAT, it means obat anti-tuberculosis. Maybe it means anti-tuberculosis drugs as antibacterial. Some people will feel queasy, and could we consume the domperidone or another queasy medicine to minimize the queasy? And please, Dr. Chikra.
Okay, thank you from Terry. It's my student in Afamedis Mandiri Sidoarjo. Thank you for your question. Can you hear me? Yeah.
Yeah, very clearly, Dr. Chikra.
Okay, about the anti-tuberculosis, like maybe rifampicin, isoniazid, pyrazinamide, streptomycin, and extra, have the some side effect. But like this one is very asked about the regime. Okay, yes, of course you can domperidone or other anxiety like this, yeah. Domperidone or metoclopramide or extra for the minimize the side effect from the tuberculosis agent. The important thing for tuberculosis is every people can use the anti-tuberculosis drugs. So everything the side effect we can use everything drugs to minimizing or minimize the side effect. So I think it's okay if you use domperidone or anti-ACT agent for minimizing for side effect from tuberculosis agent. Because the important thing is the patient can use the tuberculosis agent every time because it's during period for 6 months or failure. Okay.
Okay, that was an amazing question and answer, yeah. And this research on this tuberculosis, and I think it's very amazing and interesting, yeah, Dr. Chikra.
Yeah.
And can I ask you about something? Why the resistance can occur on tuberculosis, and how to prevent this? And please, Dr. Chikra.
Okay. Yeah, about resistance. I think it's not tuberculosis, but antibacterial extra have the possibility for resistance. As we know, bacterial resistance to antimicrobial agent is caused by three general mechanisms. The one is the first, the drug doesn't reach the target. And two, the drug is inactive or the target at which the antibiotic work is changed. Uh, we know that a failure of the drug reach the target. Outer membrane of gram-negative bacteria, but like tuberculosis, I think is the acid bacteria is barrier that can block large polar molecules from entering the bacterial cell. So small polar molecule include most antimicrobials enter the cell via protein channel like porine. The absence mutation or loss of bond can slow down the entry of for the drug to the cell of completely. Like the prevent the drug from entering porine can slow down like this. So effectively reduce the drug concentration and the active site of the drug. So if the drug's target of action lies intracellularly and the drug require active transport across cell membranes, resistant from mutation that inhibit the transport mechanism of the drug. So for example, gentamicin or which target ribosome, maybe like rifampicin in the DNA, the target. So actively transport across the cell membranes using the energy provided by the electrochemical gradient of the bacterial cell membrane. So this gradient is produced by aerobic bacterial respiratory enzyme. So a mutation in this pathway or anaerobic condition can slow down the entry the gentamicin into cell. So the value of this one is like a resistant. And then about drug inactivation. Drug inactivation, bacterial resistant to aminoglycoside and beta-lactam antibiotic usually result from the production of enzyme that modify or destroy the antibiotic. A variation of this mechanism is failure the bacteria to activate the prodrug, which is in general is the base form like Mycobacterium tuberculosis. The drug inactivation like we call in the isoniazid resistant like this. And first about change the antibiotic target, we know that like mutation from natural target like fluoroquinolone resistance, modification of occupational target like protection of ribosome from macrolide and tetracyclide, or equation of resistance from the susceptibility target like staphylococcal resistance and extra. So which is caused by the production of penicillin binding protein variant, which has a well affinity. So if we have 3K like the resistance, so we can prevent, you can get your antibiotic in the times and the good intake. Okay.
Okay, thank you for your explanation, Dr. Chikra. And we also have two questions more, Dr. Chikra, from Mr. Rio Herianto and at the first, maybe you can answer the Mr. Andri Herianto's question. And the question is, what do you think about why is it like Indonesian country is difficult to get out of the country free of tuberculosis?
Okay.
Okay, please, Dr. Chikra.
Okay. Thank you. Indonesia is a tropical country. We know that a tropical country is in the two seasons, like in rain, yeah, and then we know that it's rain have impact to the mycobacteria, I think, like mycobacteria progresses. So the important factor from Indonesia is sanitation, and then the seasons like the rainy, then and the third is maybe intake from nutrition. As we know that as we know that Mycobacterium tuberculosis can progress in the bed sanitation, and then like bed micronutrient, I think. So if Indonesia can keep the sanitation so clearly, and then can intake the micronutrient as always I think it can get out from tuberculosis epidemic. I'm sorry. I think so.
Okay, so we must keep sanitize and consume some sanitation, Dr. Chikra, and so we can free from tuberculosis in Indonesia.
Yeah, sanitation and nutrition.
Yeah, and we have come to the second, the third, the third question from Teriseta again. And if we consume metronidazole and domperidone, what time we should consume metronidazole and domperidone at the same time or domperidone 30 minutes before meal and metronidazole after meal, or metroimidazole while eating? And one more question, Dr. Chikra, from Nirasti, in the hospital where she worked, a pediatric patient is diagnosed with TB without a mental test and can be stated without further examination, physical examination by feeling the glands in the neck, or the new drug can be used for the treatment of children and can be resistant if the child is not positive or for TB, but takes TB drugs. And please, Dr. Chikra.
Okay, for Terry, I think to consume the domperidone and the metronidazole, you can consume the domperidone 30 minutes before meal, and then you can eat like this, and then you can consume metronidazole After meal, I think maybe 30 minutes too. So the consume domperidone or anti-SCT drugs can pressure or can minimizing for effect from metronidazole. So I think it's true. 30 minutes before meal for domperidone, and then 30 minutes after it for metronidazole, because we know that the metronidazole side effect is anxiety like this. Okay, and the next question, I think, Ms. Erna from Winnie Resti about the TB, like this, about TB in pediatric patient. I think for tuberculosis disease, we should the diagnose from the I think like laboratory assay like this. I mean Lowenstein-Jansen, I think Lowenstein-Jansen assay, and then MGIT assay, and then we can use laboratory assay and extra, because for tuberculosis diagnosis is important thing. We cannot diagnose tuberculosis without the laboratory assay or laboratory data or diagnosis assay, because in tuberculosis disease use the anti-tuberculosis drugs, and we know that anti-tuberculosis drug until now only to the new drug. So we cannot imagine if some people or the pediatric like this, like in case use the anti-tuberculosis without know what is the tuberculosis in their cell. So I think cannot only just visual, but needed from laboratory diagnostic, maybe Lowenstein-Jansen and MGIT and diagnostic and extra. And then for the drugs, for the TB drugs. So I think you make sure what is tuberculosis with diagnostic test, and then if we know that the tuberculosis, so we can give the anti-tuberculosis agent, and then if the have resistant, we just make sure again what is the resistant. Multiple drug resistant or extensively drug resistant or only one resistant, because we know that for anti-tuberculosis agent like streptomycin, rifampicin, isoniazid, pyrazinamide, ethambutol, so we should make sure. Okay, I think so.
Okay.
Thank you, Dr. Chikra, for your amazing answer, I think. But I'm sorry to all the participants in the sessions. We only have 3 questions, yeah, because we have limited time. And thank you for Dr. Chikra for your explanation. And we move to our second speakers, Dr. Marlon, yeah. Dr. Marlon, and here is the curriculum vitae about Dr. Marlon. Okay, and our second speakers is Dr. Marlon, and this is the profile or the curriculum vitae about Dr. Marlon. And Dr. Marlon received his doctorate pharmaceutical sciences from the graduate program in pharmaceutical science at Universidade Federal Rio de Janeiro, Brazil. And he get his Master of Science from the postgraduate program in Bioactive Product and Bioscience at the Federal University of Rio de Janeiro, and he finished his bachelor degree at the North Fluminense State University Dersi Ribeiro and concentrated in biology. And he also carrying out research activities at the Bioactive Product Laboratory and also published some papers that focus on natural products, microbiology, and immunology, and also in antimycobacterial tuberculosis. I think it's same with Dr. Chikra, right? And anti-inflammatory and antioxidant activity of extracts, fraction, and substances that obtained from plant species. And in this session, he will explain us about antimycobacterial activity from natural product. And without further ado, Dr. Marlon, you have 30 until 45 minutes to present your explanation. And Dr. Marlon, this meeting is yours.
Thank you, Erna, for the presentation. Good morning for all. Thank you, Professor.
Good morning to Marlon.
Thank you, Chikra, for the invitation.
You're welcome.
The sound is good? The sound is good?
It's very good and very clearly. So you can continue, okay. Marlon.
I share with us. For a minute. So, when it's sharing or not?
Yes, Dr. Marlon, I can see about your presentation.
Good, thank you. It's a pleasure for me to be here for talking about my project and my research and that my group developed. Yes, I'm Marlon, and nice to meet you for all. This is my email, so when I finish, when you going to send my mail, it's a question or for talking about zebrafish, antimycobacterial natural product, it's okay. So let me know. Today, I will talk not more, but a little bit antimicrobacterial activity from natural products with a focus in tuberculosis, okay? So I'm going to show you an overview of the research we have done in the last few years. Oh my God, not now, wait. Okay, so tuberculosis is one infection disease with highest mortality rate. By 2019, was estimated 10 million TB cases and more than 1 million deaths. Currently, there is an increase for the number of multi-drug resistant TB cases. This is alarming. Yeah, it's not good. In addition, extrapulmonary tuberculosis also constitutes up to 20% of reported cases. And tubercular meningitis is the most severe extrapulmonary complication. In the next slide, I will talk better about the delicate situation in between pro-inflammatory response and exacerbate inflammation. okay? But it's the dot, it's key for my research. And tuberculosis still represents a challenge in many regions of the world. For the more than 60 years, the treatment is conventional treatment with antibiotic, a combination of paracetamide, isoniazid, and tambutol and rifampicin. Yes, this antibiotic have a success rate of 86% if the patient has ideal adhesion. Yes, but if in the case of resistance, this percentage decreases down. So in case the multi-drug resistant TB, this number is 54% that success. And in case this XDR TB, this is the percentage is 30%. So it's a little. Because this it's necessary, and day by day I researching and searching for new drugs for anti-B, yes. So the TB, there are treatment. Yes, there are. The TB, there is a cure. Yes, it's possible. But there are also severe cases. For these severe cases, it's my focus of my research. Yeah. So I know, we know, although pro-inflammatory response is essential, right? But exacerbating inflammation resulting in massive tissue damage and requires specific treatment. This is very important in severe cases of TB. Because this is a one point. In another hand, the emergency resistance strengths, the high prevalence of comorbidities, and lack of success in the standard treatment lead what? Lead to expensive therapeutic regime, more complex with adverse effect and long duration. Yes, because these data are encouraging the research for new drugs, more active and with less adverse effect. Here, I put in the laser. Here, it's a note published by World Health Organization. This is talking about the necessary therapy adjuvant for help in the severe cases of tuberculosis. Yeah. So in the cases of have necrosis in the lungs, it's necessary help for another medications. For this, in searching new drugs, new medications, the natural products have an important role. Yeah. In this paper, it's a review for natural products as searching new drugs over the new four decades from 81 until 2019. Yeah. So look the graph. It's interesting data. About it's around 50% the new drugs is from natural products or derivated from natural products or inspired in natural products. It's a very important data. So among these data, nearly 70% of antibiotics are natural products or derivative from that. So look, it's 17%. It's a good number, but it's possible better. Yes. So when I'm talking about natural products, it's not only plants, but sea organism, fungi, animals. Okay. And this here is the 3 papers. I like these 3 papers because it's a review about activity of natural products against Mycobacterium. It's nice. It's so much compounds. And one paper, it's talking about anti-inflammatory and antimicrobacterial abilities. For me, it's necessary learn about this. Yes. So this report of natural compounds with antimicrobacterial activities, it's very important. So for continuing this this idea, I bring 3 papers writing for my group. Yeah, the first paper is a talk psychotrophs species from Brazilian Atlantic rainforest. It's an interesting paper because I analyzed 10 species from psychotroph, and I obtained good results. So I analyzed antimicrobacterial activity and immunomodulatory activity in macrophages, and this plant was able to inhibition NO production, also nitric oxide production, and grow Mycobacterium. In another paper, it's essential for me, is this from about fruits, this species, Schinus terebintifolium. I analyzed nitric oxide production and antioxidant activity and antimicrobacterial activity. This extract and flavonoid, it's able to inhibition again too. In the last one, this research I participated with another group. It's about seed alga, Laurencia dendroidea. Yes. From this alga, I isolated the halogenated sesquiterpenes. It's different secondary metabolites. Look, the first one it's alkaloid, the second one it's flavonoid, the third is terpenes. So it's different classes, it's possible activity. Yeah. In my thesis of doctorate, I work with Jurubatiba Restinga National Park. Yeah. Restinga, it means this typical vegetation between seed and rainforest. Yeah, it's typical vegetation of Brazil. Characteristic because it's near the sea and near the forest, so have a lot of salt. So this contribution for a new substance in the plant, maybe. Here it's me when I collect the plants. This is Restinga of Jurubatiba, where I collect the plants studied. This is the plant I studied. Look, it's more than 10, it's 12, yes. But I focus in this, the picture D from Ococha is the name, and this, the picture E, about Passiflora. I published 2 papers about these plants, and they present antimicrobacterial activity And now I study this plant. It's my focus now. Okay, this picture B, the Chiomera membranacea. This is the paper from Ocotia notata. This planting show antimicrobial and nitric oxide production inhibitory activity. So this plant was able to inhibition nitric oxide production and antimicrobial was able to grow mycobacteria inhibition. The same, the passifloricin was able to inhibition grow mycobacteria, and it was able to modulation inflammatory mediation. Yes, nitric oxide or TNF alpha, it's possible. So this is my plant now. Yes, Chiomera membranacea. But why Chiomera membranacea? Why this plant? Why I chose this plant? I went to Restinga, I collected the plant, maybe I talk with the plant, you have compounds against mycobacteria, yes, but I need confirm it have or not compounds against mycobacteria. So for this, I use same SOP methodology, antimycobacterial methodology, and immunomodulatory methodologies. Yeah, I started prepare the samples with the leaves from plant. The next step, I evaluated in Mycobacterium bovis. These strands, it's non-virulent strands because it's the first screening. So for making the first screen, it's a lot of samples. So it's needy, it's not needy, but it's good. I use it non-virulent strands. Yeah. When I have active samples, I pass for next step. I test in Mycobacterium tuberculosis. It's virulent for human. And the next step, I try for Mycobacterium marinum. Oh my God. Yeah. Then I can use infected macrophages for analyze if my compounds or extract or fraction it's able to pass in a macrophage membrane or not, and eliminate mycobacteria inside the macrophage. So it's an important analysis. And for the last one, I can test in zebrafish model or mice model if I have a good compound for test. Yes. In another hand, I can test in macrophage immunomodulatory activities. I analyze nitric oxide production, if inhibition or not, or stimulate, for example. Tumor necrosis factor alpha, TNF alpha. It's an important cytokine, yes. This cytokine is developing an important role in TB. And this enzyme, lactate dehydrogenase, it's important for cytotoxic effect. So when analyze toxic effect, I use this parameter. Look, this is my results. It's a big one table, yes? So more than 16 samples. It's not important now, analyze one by one. So look at this, the graph, and the Fox Chiomera membranacea, yeah, is my plant. This plant inhibition more than 90% the growth of Mycobacterium bovis BCG. It's non-virulent strains. And when I test against Mycobacterium tuberculosis virulent strains, I saw 97% growth inhibition. It's a good result, yes. When analyze minimal inhibition concentration for 50%, I found 4 micrograms per ml. It's a good because it's a fraction. It's not isolate compound yet. So it's a good result. It's a promiser. I analyze immunomodulatory activity in macrophages, yes, for ethanol extract and dichloromethane fraction. These 2 samples is able to inhibition NO production, and nitric oxide production, yeah, and TNF production. Look at that. When analyzed about cytotoxic effects, my plant not significant effect, toxic effects, only in high concentration, but when I try another concentration, it's good. It's not cytotoxic. It's not toxic. Because this I found in dichloromethane fraction is the better sample for the continual phytochemical study. In the next type, I test in Mycobacterium marino. Why? Because before I test in zebrafish model, I need test in Mycobacterium that I will use in infection in zebrafish model. Agree? So when analyze the minimal inhibition concentration, it's not the same. It's a little bit high, but the sample continuous inhibition. Look, this is my isolated compound, the first one. When I test 108 microgram per ml, this sample inhibition, the growth inhibition in 87%. So it's good, yeah. Because this data, I can proceed for the next step. In the next step, I try this sample, this isolated compound in zebrafish model. So for me, it's the better part of my research because it's so beautiful work with zebrafish is the special because you can see the inhibition or not. So it's amazing. This is aquarium from zebrafish. In this video, look, it's a small box. I separated male and female for the production eggs, yes? Now, look, the male and female fish have a couple fishes and producing eggs. I collect the eggs and pass for next step. This is timeline for experiment with zebrafish when I use yolk infection. When I use yolk infection, 1 day before, I need separate zebrafish in the small box, like in the last video. In the day 1, in the day 0, I collect the eggs and 4 hours post-fertilization, I infect with Mycobacterium. This Mycobacterium is a fluorescence strands green fluorescence or red fluorescence or blue fluorescence. Yes. In the zebrafish, it's transparent. So it's good because you can see the bacteria inside the fish. So the next 2 days, I incubated in laboratory. In the day 3, I analyzing COPAS machine. It's a special machine for this analyze. Thus, I started the treatment. Yes. Then I analyze 1 day, 2 days post-treatment. Look, it's interesting. Look here, the video. This video showing an infection, yolk infection. Look, it's a little big, this needle, it's little. I need the microscope for infection. I try infection and film. I make a film, so it's not good. But see, did you see? This red is the infection. So they need pass for, I can try. The needle pass for membrane and infection inside the yolk. Yes, the red, it's good infection. Not out, it's inside. After that, I put in Petri plates and analyze in COPAS. See the video. This is COPAS machine. The fish stay here in the box and pass one by one through this way. Continue, see. Look now. This fish pass for detection the fluorescence. This part. If the fish come back to Petri plate. Yes. Look now the results. The first results. The computer analyze. Yes. The blue line is the shape of fish, and the red line is the bacteria. Yes. When a line red pass for blue, it's a good infection. If not occur, it's not good. This is not good. This is not good. Yes. So after that, I analyze fish by fish. I analyze among the fish and start the treatment. It's a lot of fish, about 100 eggs. Look that. The first graphic, the red fluorescence. The red fluorescence is because I use a zebrafish line had a macrophage fluorescence. So the macrophage, it's fluorescence, red fluorescence, and I infection with green fluorescence bacteria, mycobacteria. Okay. So look at that. This is the laser here. This is a control with infected Mycobacter marinum. Yeah, it's a little high, the head fluorescence, because macrophage is recruit for the infection. This is zebrafish treatment with rifampicin. This is treatment with my ethanol extract and my compounds, isolate compound. Look, for the macrophage, my isolate compounds, not inhibition. Probably this stimulates recruiting the macrophages. So maybe this result help the organism, the fish, eliminated the bacteria. Agree? Under the under graphic is the same results, but in bar, not in dot, but it's the same. Look, this is inhibition, significant inhibition. This significant inhibition too, but for more, not for less. Yes. Another graphic, it's the green fluorescence. So it's about fluorescence mycobacteria. This is controlling not infection. So don't have fluorescence, green fluorescence. There's the next one, control mycobacterial infection. The green one, it's more, yes. When I treated with rifampicin, this decrease. When I treated with ethanolic extract, decrease. When I treated with isolated compound, decrease. So it's good result. Look under. The fluorescence mean values so high for control infection, it's green. Yes. And the rifampicin in my samples is able to inhibition grow mycobacteria. This data is significant. Yes, it's more different for compared control infection. Before that, I need to analyze another model because the first model, yolk infection, it's a direct infection. Yes, but I don't know where the infection localized inside the fish because it's only egg. Yes, so the immune response is different. It's several details. It's important. Yeah. So the next model, infection in caudal vein. Yes. I prepared the zebrafish, similar that last timeline. One day before, I prepared the zebrafish setup that is separated in micro box. Yes. The day 0, I collected the eggs, separated the eggs fertilization. And the day 1, my injection for mycobacterium is 36 hours post-fertilization. It's different. The last one is 4 hours. This is 36. So it's a little larva from zebrafish. Yes. In another day, the second day, I select the larva infected larva and start the treatment. Because I have this take care, because maybe one fish has less infection, another fish is so much infection, so it's necessary select the fish is the same profile, the infection. Yes, it's very important because it's different results in the end. No, it's not good. So I select the similar fraction and start the treatment. So I follow the treatment 1 day, 2 day, 3 day. When I finish 3 days, this is represent 5 days of zebrafish larva. Yes. I take the picture in the fluorescence microscope. Look, this is a methodology from infection in caudal vein. Yes. First, I need decolonization, egg by egg. It's an important step. The next video, look in the zone. I need to remove the chorion from zebrafish, but I don't put inside the fish, yes, because maybe hurt the fish. It's not good to hurt the fish. After that, no, not this. Look the infection. It's a complicated infection because I need to organize fish by fish in line. Look. After organization, I look by microscope and I put the needle near the caudal vein and injection occur. Look, no, not this. The red infection, it's not in one dot. Yes, it's the spray from inside the fish. It's a dissimilar infection. I think awesome methodology. Look, yes. So then I put in plate, microplate. I put one fish inside the well. I use in 24 well plates, yes. And day by day, I look for fish. It's good, it's swimming well, so the balance is well, the heart is well or not. The development it's okay or not. So I accompany this fish. And then I start the treatment. So it's nice. In finish the experiment, I analyze in this microscope, fluorescence microscope. And I obtained this picture. Look, it's a nice picture, yes. So, the first column is the fish, infected fish and not treated fish. And here, the lace. Here. The first one is Brinkfield. Yes, it's light, common light. I see the fish, transparent characteristic. So it's okay. This is a fluorescence from bacteria, mycobacteria. Yes, the fluorescence is green. So I can see mycobacteria inside the fish. So a lot of dots, the bacteria, the fluorescence. And this, when analyzed red fluorescence, it's talking about macrophages. Yes. And here is the merge this tree picture. Yes. So look, I can see zoom. No, yes. Here. Nice. Look at that. The vesicle, it's extravesicular, yes, have macrophage and mycobacteria. This is like to granulome in tuberculosis, yes, because fish it's not have lugs, but the granulome it's like this. It's extravesicular with mycobacteria and macrophages, yes. Oh my God. Yes. Next. So here, the second column, my fish treated with my compound. Look, the group treated with isolated compound. The dots, it's small. I can see dots green, yes, but less than infected fish. Look, it's nice, yes. Just suggests that my isolated compound inhibition mycobacteria. Agree? The next here, it's red fluorescence about macrophages. Look, it's have fluorescence macrophage probably because my sample help recruit macrophage for eliminate mycobacteria, probably, yes. And this, it's not formation granulome. It's nice, yes. And another point I need to highlight here, look for the fish 1, column 1, and look the fish with treatment. Look here, the eyes, the fish not have balance. Look, this eyes, it's under 1 and 1, the eyes, yes. In this fish, it's not because she lost the balance. So it's moved. Another dot is about it's swim. I don't remember the name. It's necessary for balance. So this development is natural development. The circle black, yes. So here it's not development. It's difference. So tuberculosis in fish, it's the fish it's suffer about mycobacteria. Yes, I understand. It's so confused. Yes, but in the finish the lecture, it asked me. I explain better. Yes, but my fish demonstrating healthy fish. This it's not healthy fish. This is an important point. So I can change this picture for a graphic. Yes, this is the graphic. So control infection, a lot of fluorescence, around 100, and this is the treatment. So my isolated compound and rifampicin in the same concentration, 32 micromolar. Yes. Look, here it's only almost nearly 18% of fluorescence, and rifampicin 12% of fluorescence. This is mean just 82% the inhibition and 88% of inhibition. When I try rifampicin in high concentration, I obtained 97% the inhibition. So it's a good result because this is isolated compound from plant. Yes, it's a simple analyze and a good result. It's a promising result. So I start in vitro analyze in prostate. It's next step. I arrived in zebrafish. So I obtained good result, promising for a new drug about this compound. In conclusion, natural product samples show promising activity against Mycobacterium growth. The isolated compound from Chiomera membranus was activity against Mycobacterium growth in zebrafish larva infection model. Was possible to demonstrate that in vitro screen may be an efficient strategy for select bioactive samples. Yes, because the bioactive samples in vitro is the same in vivo zebrafish. Yes, and the sample, the compound, isolated compound, it's a good sample. It's a good compound. Probably I follow in murine model, mice model. Yes. So I grateful the situation from Brazil. Is a lot of institution and my university and my sandwich in Leiden University and my group here because I don't make this alone. This is a group. Thank you, my supervisor, Professor Michel, and my supervisor Fons in Herman in Leiden in Netherlands, and in this another students participation with me the research. Yes. So thank you again, terima kasih and obrigado in Portuguese. Thank you.
Okay, Dr. Marlon, that was an amazing presentation, I think, yeah. And your research is focused also on TB, like Dr. Chikra, yeah, I think, but Brazil is lucky, yeah, because Brazil has Chiomera membranaceae that you explained to us that it has an active compound, and you name it compound 1, right? And it has an activity that against mycobacterial growth. And in this session, Dr. Marlon, we have to come to question and answer session, I mean the discussion session. And in the chat Zoom, we have...
Bentar. Coba konfirmasi ke Marlon, udah dengar belum?
Can you hear me, Dr. Marlon?
Yes.
Yes, clearly?
Yes.
Yes. Okay, thank you, and I will continue. And in the Zoom chat, we have 3 questions from student of Akademi Farmasi Mitra Sehat Mandiri Sidoarjo, and there is the 3 questions from Mr. Haikal, and then Miss Kurotun Aini, and Mr. Terry Septa. At the first, I will ask you about the question from Miss Kurotun Aini. And the question is, how about the natural product mechanism as anti-tuberculosis agent? How about the natural product mechanism as anti-tuberculosis agents? And what a dose maximum? Maybe what is a maximum dose for it? And thank you, Dr. Marwan. Please answer the question.
Okay, thank you for the question. So have many mechanism action. Yes, maybe interaction from cell wall or disturbed cell wall. And in specific, my compound, it's interlink interact with protein tyrosine phosphatase. Yeah, you know, this is responsible for virulence in mycobacteria. So my compound interaction with this and inhibition. So it's a one mechanism anti-mycobacteria. Yes, but I have another mechanism. Each one natural products have one. Yes, it's different mechanisms. So it's specific. It's necessary analyze for one by one products. Yes, and the dose maximums. It's important analyze for cytotoxic effect, toxic effect in zebrafish. So the same the last response. It is specific for the each one compound. Because this is necessary, very important. I analyze the toxic effect for each one compound. Yes, so I don't bring it now for my toxic effect in zebrafish, but my compound does not show a toxic effect in zebrafish. It's important because this is activity in 32 micromolar, but in 32 micromolar, it's not toxic. So I need to try high concentration for I found toxic effect. Yes, but I tested until 512 micromolar, and I don't found toxic effect in my compound. Yes, but each one compound it's necessary this evaluation. It's okay.
Okay, thank you, Dr. Marlon, for your answer. And as I can conclude that we need to study more again, yeah, Dr. Marlon, about the maximum dose of your natural product. I mean, your compound one. And we have come to the second question from our student from Akademi Farmasi Mitra Sehat Mandiri Sidoarjo, Mr. Terry Septa, asked you about how long we could prepare an activities of nature material until we can justice that can be an antibacterial agent. And that is the question. Okay, Dr. Marlon, please answer the question.
How long the time is I need for prepare my extract? Yes. Okay. So I start the prepare my material, and I think it's 1 month. Yes, for I found in crude extract. Yes. Then I prepare the fractions with exam ethylacetate and dichloromethane butanol. It's different solvent, different polarity for extract different classes of compounds. Yes. So I needed 6 months about this for prepare the extracting fraction. But I had look in my research, because when I tried the first column, the column for separate the compounds from fraction. I have one fraction from dichloromethane. Yes, this fraction is a mix of compounds, so I need separate one by one compound. I'll try this. Yes. So in the first column for this methodology, the class column, I use cephadic silica for this separation. In the first time, I found I get 100 milligram from my compound, isolated compound. So when I know about natural product, this is a lot of natural products about isolated compound. Normally the mass it's a little. So the first one I get 100. It's good because this I try in zebrafish test and mice test. Yes. What the question? And
Okay,
in my
Dr. Marwan. So it needs almost 6 months to prepare it.
yes, 6 months. Yes.
Yeah, 6 months, and it's also difficult, yeah, Dr. Marwan, to isolate new compound. And I'm so interested.
Sorry, because I had a good look. Because I had a good look.
Yeah, yes, that has a good look. Okay, and I'm so interested about your research, Dr. Marwan. Can you show us about the structure of the compound one?
Yes.
Or maybe it, yeah.
I know the compound. I know the structure for the compound, but it's complicated. Said the name. Talking about the name. Yeah, because it's not published yet.
Okay, that was
it's a flavonoid class. It's a one flavonoid. But this flavonoid, it's a methox group. So this methox group in flavonoids make the flavonoid less polar. Yes.
Yeah.
When this occurs, it's facilitated for compound pass for cell wall.
Okay, Dr. Marwan. Okay, thank you. I'm so interested with your research. And now we've come to the third question, Dr. Marwan. This is from Mr. Haikal and also from Akademi Farmasi Mitra Sehat Mandiri Sidoarjo. But this is, and the question is how to treat psychotherapy that is effective for patients with TB. Okay, please answer, Dr. Marlon.
It's a good question. Yes, phytotherapy for the tuberculosis. It's a good question. And a complicated question. Because natural products, the mass of products, natural products, is small. Yes. So for phytotherapy against mycobacteria, it's necessary another study. For example, I can use extract from the plant in not isolated compound. Yes. So I need to study how I prepare this phytotherapy. Yes. How I analyze the amount compound inside the extract. And I need a stud in the farm, a farm for the plant, for prepare this phytotherapy. So it's delicate because natural products, yes, but it's possible if had these experiments, these studies about farm of plant, about the necessary amount in the substance inside the extract. So it's necessary for a recommendation phytotherapy for mycobacteria. But the isolated compound from natural product, it's important because you need for inspiration and synthesize substance in lab. Yeah. So this is possible new drug, for example. Yes. Maybe not for phytotherapy, but maybe not it's inspired and make the synthesized compounds. Yes. But interesting questions. Yes.
Okay, Dr.
In Brazil,
Marlon,
it's half
because
complicated.
we have a limited time. Yeah, also complicated. And because we have limited time, we have come to the next question, ya, Dr. Marlon, from Miss Fatima. She has 3 questions, but I think the first question and the third question is done. And I will ask you about the second question. Why the zebrafish assay use Mycobacterium marinum, not Mycobacterium tuberculosis? And I'm also interested with your animal test, yeah, because in the past we just only use like a rabbit or sometimes rat like that. Why you are using zebrafish as your
Experiment.
test animals? Okay, please, Dr. Marlon, please answer the question.
So I use a zebrafish because it's a fast model, it's an easy model for natural products. Because I could look for isolate compound, but normally the isolated compound it's a few, it's a little bit, yes. And in zebrafish, it's necessary a little bit simple. So it's good for natural products because this zebrafish, yes, it's a fast test. So in 5 days, I have the results. It's fast, yes. When I thought about mice, I need 30 or 1 month and a half for analyze the mice. Yes, so a long time, and I need more samples. Yes, more samples. Okay, I use Mycobacterium marino, not tuberculosis, because marino is natural host for zebrafish, so it's easy for infection. Yes. Maybe I can use tuberculosis for infection zebrafish. Yes, but it's necessary high level biosegurance. Yes, and I need to have a first result in Mycobacterium marinum, because if my sample not inhibition Mycobacterium marinum inside the zebrafish, probably not inhibition Mycobacterium tuberculosis. Yes, it's probably, it's not it's probably, it's inhibition, yes, because this.
Okay. Okay, as I can conclude that you use zebrafish because it just only need a small dose, I think, yeah, from your isolated compound one, and then it give us or more faster than mice, as I can maybe you can show the reaction like that.
Yeah.
And that was
the research, my research involved inflammatory profile. Yes, so I can see in zebrafish this profile because I can see recruiting macrophages. I can see analyzing cytokines inside the fish. So I can see analyzing EL1 beta, TNF alpha inside the fish. So it's good for me because my isolated compound is dual activity, anti-inflammatory and antimicrobacterial.
Okay, Dr. Marlon, that was an amazing answer, yeah. And there is Dr. Chikra raise her hand, and maybe Dr. Chikra, can you join the Zoom?
Yeah, okay, thank you, Miss Erna. I think about Mycobacterium marinum and zebrafish is close interest because the Mycobacterium marinum in zebrafish can give the macrophage like this, and then we can know about the mechanism of action from the natural product or synthesis or extra in the effective times more effectively. And then we can know about the other mechanism like LC3 in zebrafish use the Mycobacterium marinum. So if we use Mycobacterium tuberculosis in zebrafish, I think we cannot know or we cannot see about the macrophages and extra or the immunological type in the zebrafish. So if we use zebrafish, should use the Mycobacterium marinum, not Mycobacterium tuberculosis.
But Chikra, have a paper that show the Mycobacterium tuberculosis infection in zebrafish too. Yes.
Yeah.
I don't
Maybe
know
I think
what
it's
is analyzed.
little. Yeah.
I don't know what to
but
analyze.
I think okay, but for the tuberculosis effect, I think is we can know about the macrophages from Mycobacterium marinum in zebrafish. So if we use Mycobacterium tuberculosis in zebrafish, it's okay. But we don't know about the macrophages in the tuberculosis disease. I think so.
Yeah,
Thank you.
I agree.
Yeah, I think we have agree because our research is same in the same laboratory in the same zebrafish, so we have the same opinion. But I think it's if we use Mycobacterium tuberculosis and zebrafish, maybe for other disease or other parameters in tuberculosis disease. But until now, we read. I think I read in the paper. Only Mycobacterium marinum can describe how to the progressivity in tuberculosis disease. But other parameters, I don't know. So maybe in other paper on other sometimes we can learn more deep again for Mycobacterium tuberculosis in zebrafish. Thank you.
Okay, thank you for Dr. Chikra and Dr. Marlon. Because we have limited time, so thank you so much, Dr. Marlon, about your interesting and amazing presentation today. And now, ladies and gentlemen, we have come to the third speakers, Mr. Joao also from Brazil, and I will deliver you to the curriculum vitae of Mr. Joao. This is the profile of the Mr. Joao, and Mr. Joao is a graduate student, I mean, from pharmacy at Universidad Federal Rio de Janeiro, Brazil. Same with Mr. Marlon, yeah, I think, yeah. And his research is focused on mutagenicity of compounds. And in this conference, he will explain to us about the AMES test, a way to evaluate the mutagenicity of compounds. And Mr. Joao, you have 30 to 45 minutes to explain about your presentation. Okay, Mr. Joao, and this meeting is yours.
Okay, thank you for the presentation.
Ini lebih cakep.
Can you guys see the presentation?
I'm sorry, Mr. Joao, I cannot hear your voice clearly. Mr. Joao, can you hear me?
Ini masih lebih ganteng lah. Ya kan?
Can you hear me?
Hello, Joao. Can you hear me?
Yes, I can hear.
Yes.
Just a-
Okay. I can
Okay.
hear you very clearly because Miss
You can
Erna.
hear me?
Okay. Yeah.
You can hear?
You
Oh, I
can
think.
continue for your presentation. It's time
Yeah.
for you. Yeah.
Can
Welcome, Joao, in Indonesia.
Oh, thank you. Appreciate it. So glad to have this
Okay.
opportunity.
Yeah.
So can
Okay,
you
you can
so the
continue about your presentation.
okay? Can you see?
Yes, Mr. Joao, I can see about your presentation and your voice is so clearly. So you can continue about your presentation.
Okay, thank you. So my name is João. I'm a graduating student of pharmacy in UFRJ. It's a college in Rio de Janeiro in Brazil. So I'm gonna present a little bit of my research, a little bit of the AIMS test, and it is a part of my research. So we already know how long and expensive is the process of discovering a new drug, and it was necessary about 10,000 compounds for have just one drug in the final of this process. And so everything start in a screening for a substance for certain potential, for example, anti-inflammatory potential or antiviral potential or for the anti-TB potential. And in this process, to make this process the more efficient as possible, it's important to start the standard security test in the beginning of the preclinical stage. So the preclinical stage is you is using a severe test for like pharmacological tests or pharmacokinetical tests and the toxicological tests too. So this toxicological test is made to prevent risks and reduce the probability of these compounds fail in more advanced steps, like you so you reduce in a future test, after a lot of time and money expended in this compound, and imagine you discover this is toxic and in, uh, in another expense part of the process. So with there's some tests of security you have to do, like for example a single dose test, or multiple dose test, or toxicity for reproduce function, and the genotoxicity test we are going to talking about. So this genotoxicity test is made to prove information about the potential of these compounds to induce DNA modifications. So so basically, if these compounds had the potential of mutational or not. In the AIMS test is one of those genotoxic tests that we widely use around the world, because it's a simple and an expensive test for genotoxic for compounds. So the AIMS test was invented by Bruce Ames in 1970, using the strains of Salmonella typhimurium. The test is based on the capacity of the compound reverting a pre-existing mutation. So these strains have a pre-existing mutation in the DNA who it doesn't produce histidine. This is essential for the growth. And the FDA, the Food and Drug Administration in USA, they recommend this AIMS test, but it's not required for you present those results. But the MDA, the European Medicine Agency, describe two options for this standard test, and both options, the AIMS test is required. You are obligated to show the results of those AIMS tests. In Brazil, we have this. The FDA of Brazil is called Anvisa. They regulate this type of experiment, and like the EMI, the Anvisa describe two options of those standard tests, and in both of them, the AIMS test is founded. So the first option, we have the AIMS test, a test for gene mutation in bacteria, and a cytogenetic test for chromosomal damage, and in a vivo test for genotoxicity. Generally, generally they use for chromosomal damage in hematopoietic cells from mouse. In the second option, we also have the AIMS test and two in vivo tests. One of toxicity of using a micronucleus, whose hematopoietic cells, and another test in vivo using a different test. So does the results of a test in bacteria have any significance to in detection of toxicity for humans? Like the genetic and the chemical nature of DNA is identical in all organisms. The genome is equal for all organs, and aims develop a way to simulate the metabolism of the human metabolism in the bacterial systems. So in general, in mammals, we process the drugs happen in the liver with by some enzymes. And in some case, the action of those enzymes can can create a toxic or mutagenic compound. So these metabolism can generate a mutagenic metabolites. So, and the aims also create different samples of Salmonella typhimurium to detect different types of mutation. So we have all these strains, and the most of used is TA100 and TA98 and TA102. The TA100 have a histidine gene that promotes a replacement of a leucine with a proline in DNA and thus detected the mutation of the pair base pair, base pair substitution. So when you had this test, we have a different way to the compounds make the mutation in the DNA. So we have the base pair substitution and the frame shifting. That's the different mechanisms of reaction of mutation. So different strains can detect these different mutations. And the frame shifting mutation is affect the correct reading of a codon sequence. So it's a different mechanism of reaction. The strains also have another mutation uh, that make the test easier. So, and we had these mutations is important. It's important to make a quality of control for this mutation, because if you doesn't have these pre-existing mutations, the test is gonna be wrong. So it's important to have a quality control of these mutations. So in for the exotrophic histidine, the cells doesn't produce histidine because they have this pre-existing mutation, and the test is based on a compound revert this mutation for can cell have back to produce histidine. So we have uh, two two plates, one with histidine and another without histidine. So the strain only should grow in the plate who has histidine. So like you can see here, this plate you didn't add histidine, so you can not see any growth of cells, any bacterial growth. And in this place you had histidine, you saw this bacterial growth. And you also gonna see this division in the plate. It is those are division are made for all quality tests, because we have different 5 different stocks for the same strain. For example, for TA98, you have 5 TA98 strains, and we will test all together to compare and choose the best one to make the final test. So you in all those control test, you're gonna see these lines to separate these strains. This RFA mutation is a mutation in a bacterial cell membrane. So which makes the membranes more permeable for large molecules, so for large compounds. So we use the violet crystal, the substance, who is a large compound that can only kill the bacteria if they enter in the cell. So if the mutation existing in the cell, you're gonna see these. I'm sorry. You're gonna see this zone, this inhibition zone around the paper, who is full of violet crystal. If they doesn't have this mutation, you doesn't gonna see this zone. The bacteria grow around the violet crystal. Because imagine you are testing a substance and a big substance, and they doesn't enter in the cell and doesn't uh, reach the DNA. So you have a false negative result because the substance cannot uh, reach the DNA. So of that, this mutation is important. So the deletion of URVB gene is important too, this exclusion of this gene, because the DNA repairs system in those cells should not happen. So if those cells have this mutation in the gene, they responsible for the proteins who responsible for repair the DNA damage. So in this test, we irradiate a half of the plate. This is non-irradiated part and this irradiated part with the URV, the URV radiation. So if the this URVB radiation will cause damage in the DNA. So if the cell is able to repair this damage during the incubation, there will grow bacteria around here in this side. So if they doesn't repair this damage who the UVB radiation makes, it doesn't grow. So you can just see a little of grow here, but not compare like the non-radiation side. And the the bacteria also have insertion for a PKM 101 promotes ampicillin resistance for those cells. So it's important to know this. There's only those strains grow in the plate. There's no other bacteria grow. So this insertion of this plasmid promotes ampicillin resistance. So we can see in those lines, we add ampicillin in the plate, and we saw bacteria grow in there. And when you add another antibiotic, for example, tetracycline, we saw this inhibition in the lines. So the bacteria doesn't grow. And the the test itself is very simple. So it's for each concentration you done in triplicate. So if without and with a mix of enzymes that can that this you call this mix of enzyme for S9. And in the test tube we add the strains of salmonella and the sample and this mix of enzymes or not, and pre-incubate for 30 minutes to for can the enzymes and the sample can react with the strains with the cells. So after that we put on a Petri plate, then incubate for 72 hours for the bacterial growth. So if if the compound, the sample, uh, can have induce the mutation, we saw a lot of uh, cells in the plate. And if it doesn't induce the mutation, you just have some bacterial growth because a spontaneous mutation is expected. So in the real plate, we saw the negative control, so it doesn't have any substance, just the spontaneous the, the spontaneous growth, the spontaneous reverted mutation grows. And here we saw the positive control, like when the substance can can mute can induce a mutation, you had this clear difference in the plate. And these other plates is from the sample we test, and you saw that induce a mutation. So we can calculate the mutation rate. Is one way to quantify the how can the sample can induce a mutation. We can quantify this. So if in a simple mathematics, we just we just uh, divide the number of columns reversed on the plate who add the sample by the number of colonies who in the negative control who have a spontaneous reversals. So if the mutation rate, the MR, is equal to 1, indicates there is not induction of reversals. So it's not a mutagenic compound. If the MR is less than 0.6, indicates of toxicity for the cells. And if the MR is more than 1, this may indicate a induction of a reversal that can be mutagenic. So, and if the MR is equal or superior to 2, the substance is considered mutagenic. So it's an example for results, not my results. It's from this paper here. And it's interesting because for the question, we saw they have a induce the mutation. It's a mutagenic compound in those concentration of 24.5 nanomoles per plate. We already saw the mutagenic. Is the MR, the mutagenicity rate, is more than 2. We saw this in without the mix of enzymes and with this mix of enzymes. With the mix at the first concentration, we saw that is mutagenic, and we saw that is 20 times than the negative control. So it's really induced the mutagenic is really mutagenic. And for the cainferol, we didn't saw any mutagenic without this mix of enzymes, without the S9. With the S9, we just saw the mutagenic with this mix of enzymes, who we can say that the cainferol is only able to cause the mutation with S9, with indicating this flavonoid when he's metabolized by liver, that generates metabolites that is capable to cause mutations. And we can also say that cainferol, the mechanism of mutation for cainferol, like for the TA100, that you cannot see any mutation, you just can see in TA98. So the mechanism of mutation from cainferol is that frame shifting mutation. The reading code, the starting reading code, it's wrong, and that's the mechanism of this cainferol mutation. So thank you.
Okay, thanks for Mr. Joao from your interesting and amazing explanation about AMES test. And now, to all of the participants today, we have come to question and answer session or discussion sessions. And we have 10 until 15 minutes to discuss about this. And maybe you can write down your question on your chat box in the Zoom. And Mr. Joao, based on your explanation today, your research is focused on AMES test, yeah. Test for gene mutation in bacteria, right?
Yeah, exactly.
Yeah. Can I ask you about something, Mr. Joao? The first question is, why are you use AMES test with bacterial, and what is the benefit of this? Okay, thank you, Mr. Joao. Maybe you can answer about this question.
Yeah, you use bacteria because the facility and the speed of the test in comparison with the in vivo test, like in mouse test, is really fast and inexpensive. It's a cheaper test. So, and also we didn't use animals for that. So we use the bacteria because the facility, and we have a good results. We can also saw the same results in vivo tests, and the test previews mutagenic compounds in vivo too.
Okay, Mr. Joao, as I can conclude about your answer, yeah, I mean, you use AMES test because it cheap at first and has a short time analysis like that,
Yeah.
and maximal in your research, I think. And Mr. Joao, we have a question from our student from Akademi Farmasi Mitra Sehat Mandiri Sidoarjo. And the first question is from Mr. Terry Septa. And the question is, the strength of flavonoid could be an antibacterial for TP, I mean Salmonella typhosa, and maybe in maintaining health before the infection of Salmonella typhosa. And okay, Mr. Joao, please answer the question.
So the this flavonoid we saw in these results is not from my results.
Yeah,
So,
uh,
but
Mr.
yeah,
Joao, there
the
is
question is if those compounds can is toxic for cell and doesn't see any grow in the plate, things like that.
Uh, he means not flavonoid, Mister, but quercetin. He means.
Because I didn't understand the question. Sorry.
You didn't understand the question, so I will read again about the question from Terry Septa. The strength of quercetin could be an antibacterial for Salmonella typhosa. Maybe that the means of this question.
Okay. Because the kerosene can kill the salmonella cells. So it happens. So we test have a toxic for cells test before we made this test. So we we made the test with the concentration that cannot kill those cells, because if the compound is toxic for the cell, we can see any grow, and you can analyze the mutagenesis for these cells. But when we analyzed those compounds, we had these antibacterial activity, we had this problem. So with you can also make in other simple tests like the AIMS, but we usually made the AIMS test with a concentration that you cannot kill those cells, just induce a mutation.
Okay, Mr. Joao, thanks for your answer about that. And we now move to the second question from Miss, wait, from Miss Siti Aisyah. This is a simple question, Mister. She asked you about what is the conclusion about your research. Maybe you can explain to us in simple ways.
So the results not that I show is not my results. So I made these AIMS tests for just one sample, because the objective of my research is to make for many samples, like made for shell cones. So where does the pandemic I can do this test for others shell cones, but I made just for one for now, and this cell cone test is not mutagenic. They can present the mutagenic activity. Is not mutagenic compound that I investigated.
Okay, that was amazing, yeah, amazing research about AMES test for gene mutation in bacteria. Uh, Maybe Dr. Chikra, she raised her hand. Maybe that's something. Maybe Dr. Chikra can join to the Zoom discussion.
Okay, thank you, Mr. Erna. Joel, can you close your presentation,
Yeah, so
please? So I can
yeah,
see you very clearly.
okay, thank
Okay.
you.
Thank you, Joel. Okay, I just want to say about Terry's question about quercetin. Yeah. Yeah, interesting question about quercetin. Quercetin is flavonoid groups, I think. Uh, it is have uh, higher antibacterial activity because it is have hydroxyl group, so it can inhibit, and maybe it's some paper said about quercetin can kill the bacteria. And then how about the treat if we use quercetin in the health? I think, wait, I will read from Terry. How about if we use flavonoid, if we use quercetin as maintaining health before infection Salmonella typhosa? I think maybe there is answer if we use quercetin before we infected. I think it's a natural product can use as preventing agent. So if we use quercetin before we infected, if it's better because a natural product is prevent from infecting antibacteria, and then a natural product we can use as the maintenance product. So if we use before kill or before infected is more better. So I think that, and then about the Joao research, multigenetic assay with bacterial method, it's more effectively, and in Indonesia is little little institution or little paper submitted about the mutageneity test with bacteria. So maybe we can join for research together about mutageneity assay with bacterial method, like your research. But I will ask for you about the mutageneity assay with the bacterial, only bacterial gram negative or gram positive, or how specification about the bacteria can use the mutageneity assay. Thank you.
So in the test, the protocols I mentioned, we is not mentioning any of the use of the different cells, different types of salmonella or for different cells, but they just use the salmonella typhimurium. But we can also use E. coli cells too. We can use E. coli strains, but it's
Okay.
not common. It's
Oh,
just
okay.
common to use
Yeah.
those salmonella strains. But
Uh,
we have this strain.
okay. So I think if we use Salmonella TP, have a high probability for mutagenicity test like this.
Yeah, it's easier to mm, to conserve those pre-existing mutation for the test. Like
Okay.
the bacteria is so sensitive, like they doesn't produce histidine, they have
Okay.
mutation of in this membrane in the
Okay.
DNA repair system. So these salmonella, it's easier to conserve this mutation we have
Oh,
to
okay.
have the test.
Okay, great. Thank you. It's perfect experience for me.
Thank you.
Okay, thank you for Dr. Chikra, and thank you for Mr. Marlon. I mean Mr. Joao for your such nice presentation explanation about anastas for to us. And because we have limited time, Mister, and I would like to thank you to Dr. Marlon and then Mr. Joao and Dr. Chikra for such nice and interesting presentation today. And me as moderator hope that the information that delivered from Dr. Marlon, Mr. Joao, and Dr. Chikra will give us a benefit. And okay, that was an interesting session. And because the time is up, and I will return the time to the MC.
Thank you, Mr. Ms. Erna Fitriani, Master of Science, who has guided the material presentation and discussion in this AIC today. I would like to thank you to Dr. Marlon, Mr. Joao, and Dr. Chika for such nice and interesting presentation today. The next event is the reading of prayers by it's by Mr. Rahmat Farisi. Mr. Rahmat Farisi, this time is yours.
Yes, thank you. Assalamualaikum warahmatullahi wabarakatuh. Sorry, I am informing that this prayer is specifically for Muslims. Non-Muslims, please pray according to your respective belief. Before we pray, let's us bow our head for a moment, clear our mind, focus our heart on our, so that our prayers are accepted by Allah Subhanahu wa Ta'ala. Bismillahirrahmanirrahim. O all merciful Allah, thank you for your mercy and compassion, because all our activities at this international seminar run smoothly, even though it requires time, effort, and truth. And today, our Lord thank us close our activities to our bless all of our activities while following our series of events and make it a form of worship by yourself. For that, O Allah, give your mercy and compassion to us and make this meeting a meeting that you are blessed with. O Allah, open for us the door of goodness, the door of your grace, the door of your strength, the door of your faith, the door of help, the door of salvation, because you are the most gracious and merciful. Please pour out, please use out, keep our stand again temptation. O Allah, please give all the knowledge we got during this activity so that we can practice in the future, so that we can become role models or the family, community, national, and state. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار سبحان ربك رب العزة عما يصفون وسلام على المرسلين والحمد لله رب العالمين وبالله رب العالمين. Thank you.
Well, ladies and gentlemen, finally, we come to the end of webinar conference. We would like to say thanks again to the all for the presenters for their informative and interesting talk, and to audience for your active participation. Thank you for your attention. Good afternoon. See you the next time.
Thank you. Thank you for Marlon and Joao. It's time for sleeping for you all. For Brazil's time at I think 2 a.m.
1 a.m.
Yeah, okay, 1 a.m. So you can go to sleeping. Okay, thank you. Many thanks for Marlon and Joe. See you later. And you should go to Indonesia, to Sidoarjo, and to more holiday. Once again, thank you for sharing knowledge and your time. Maybe we can to be continue soon in WA group, maybe. Oh, I'm sorry. Nggak ada foto bersama kah? Or maybe Marlon and Joe, can you still hear?
Yes.
Yes.
Okay. Maybe Joe, can you open your video to get
Yeah.
take a photo, take a picture together.
Yeah.
Yeah. Joe, you look so handsome, and many students from Mitra Sehat Mandiri Sidoarjo say that you look so handsome.
Thank you.
More handsome than Marlon. Okay, so do you have a girl's friend?
No, it doesn't have a girlfriend.
Okay. Okay, siapa yang take picture? Okay. Okay, please open the video for all audience, all members. We will take pics together for our memorize. Okay, Mr. Erna, can you guide us? Okay.
Okay, to all of the participants today, and for Dr. Marwan, Mr. Joao, Dr. Citra, and all of us, please open the camera and I will take a picture for us. Okay, I will count until 3 and you can give us your best. 1, 2, 3. Okay, for the second page in the Zoom, One, two, three. And for our third page, 1, 2, 3. For the fourth page, 1, 2, 3. And for the last page, please open your camera and I will take a picture for us. And I will count until 3. 1, 2, 3. Okay, Dr. Marwan and Mr. Joao, Dr. Cikra, and for all of the participants today, thank you for joining us in this AIC conference, and maybe we will, or I think maybe we can discuss in another. See you.
Okay, see you. Bye, Marlon. Bye, Joel.
Thank you.
Thank you.
Thank you. Bye bye.
Bye-bye.